Good morning, friends. Myself, Major Dr. M. Q. Beg. You are viewing the Major Dr. Beg Oncology classes. In connection to that, my new topic of presentation is concept of molecular oncology, precisely in the oncology practice. Why it is important and how much this is important for an oncologist to understand for the new era of oncology treatment, the molecular oncology. So, I have made a very simplified version of molecular oncology slides, and for your teaching, since it is a very important lecture, so that each and every slides is important, and what I am telling you must see this thing. So, why molecular oncology is important in clinical oncology practice in coming era? Because it helps to understand the cancer initiation and growth as well as cancer progression. The understanding of these will promote the development of specific pathway targeted drugs, which we are practicing now. So, for for practicing targeted drugs, you really can't avoid the molecular biology and oncology for treatment purpose. A few of the cancers are hereditary and familiar in nature, that by the help of molecular oncology and molecular biology can be picked up by the help of genetic counseling, and several tests are available. But for that, your molecular oncology understanding is very important. There are future trials also required to collect the data of a homogeneous molecular group of patients, so that you have a pool of patients, and then for a homogeneous molecular group of patients you have a homogeneous drug specifically designed for that, and so that you can blindly prescribe these drugs in those group of patients data is being collected. So this is a very important message in the molecular oncology. So, before embarking on the details of the cancer molecular oncology, we should understand some basic things. The cancer cells, those cells that can overcome the boundaries of impending unrestrained division, will multiply. The cancer cells sustain the opportunity to acquire further aberrations that fuels its growth, survival, invasion, and migration, that is metastasis, to establish its presence in the distant organs. The cancer also is a clonal disorder, because there is evidence that most of the cancer cells arise from a single clone of a cells, and that clone has again got disruption in its regulatory mechanism, and then lead to the proliferation, and then lead to the proliferation. So that is the uh, chronogen cells responsible for the cancer development. Why malignant cells are different from a normal cells? The cancer cells often grow more rapidly in compared to the normal cells of a tissue of origin. The second is the cancer cells fail to show normal cell-to-cell interactions. Normally, the cells interact with each other for the diffusion purpose, for that there's so many purpose of in physiological purpose. But when the cells-to-cell interaction is lost, the cancer cells becomes independent and starts ability having acquire the ability of to invade the in local tissues, nearby tissues, and as well as metastasize. And also that invaded cells, those cells whose seedlings went through the blood or lymphatic channels, also have ability to develop, to grow in a foreign environment, and also develop the neovascularization, that is called angiogenesis, and to develop in the foreign environment. Usually we see the cancer cells are immortal, or at least have ability to divide many more times than compared to the normal cells. So what is the capacity of a normal cells to divide? That is almost 50 to 60 times. After that, the significant portion of a telomere is lost, and the cancer cell lost their proliferations. Normal cells lost their proliferation, but that that there is a telomere is shortening is not happening in the cancer cells. So that's why, because of certain enzymes and some problems are there, so that cancer cells becomes immortal and then keep on dividing and dividing, and they does not go for a normal apoptosis, that is the programmed cell death. So what are the external agents are responsible for the development of a cancer? That's some ionizing radiation, chemotherapy, that's chemo induced DNA damage. There are certain oncogenes and some viruses and telomere attrition. So these are the factors which are responsible for the DNA damage, and then as well as the DNA damage lead to the some mutations in that I'll be coming to that, and that lead to the development of cancer. The mechanism of carcinogenesis is a multi-step process with a multiple genetic alteration occur over extended period of time. Sometimes these genetic alterations occur at the germline level. For example, the cancer predisposing syndrome such as retinoblastoma, but the most of the alteration occur in form of somatic mutation. So they are, I mean, you got a two clear-cut message that cancer cells development occur by the somatic mutations or a genetic alterations. So the somatic mutations is a dominant phenomena. Most commonly, more cancers are developed because of somatic mutations. There's also chromosomal translocation is responsible for the development of cancer. The deletion, a point mutation in a chromosomal translocation with a particular chromosome segment is having an oncogene is to get translocated somewhere else, and that proto-oncogene is translocated to other segment of a DNA that becomes activated, and that behaves like a that proto-oncogene as an oncogene and lead to the cancer. So the point mutation is also the one part. The proliferation is controlled by the sequence of signals affecting the cell's division and a cell differentiation. Signals may be positive or a negative type. An occurrence of tumorigenicity is a result of a change that affects the control point of that positive or negative. That I'll explain this in this slide. You see the gain of function of a mutation in oncogene, that is BCL2 gene. BCL2 is supposed to be oncogene, that is also called anti-apoptotic gene, the responsible for the upregulation and result of a cancer progression. But another gene, for example, there are several genes, but another gene which is called tumor suppressor gene, that is P53, if a loss of a function of this tumor suppressor gene P53 or some mutation in that lead to the cancer progression. So that happens in the two way. So let us talk about some oncogenes. Oncogenes were first discovered from the study of retroviruses, and precisely saying the rhesus sarcoma virus was then with the first retrovirus is identified causing the cancer, and this causes a cancer in animals caused by the retrovirus mediated through cellular genes that has been mutated. The normal version of these genes, that is called oncogenes, is lies in every mammalian cells in form of proto-oncogenes. So that everybody is having that proto-oncogene, but it is not harmful at all. But when the mutation occur in this proto-oncogene, some by the external insults, which I have already told, it may be radiation, chemicals, ionizing radiation, chemicals, and viruses, that lead to the conversion of proto-oncogene into the main oncogene, and this is responsible for the development of cancer. So that I've already told you the various agents and the factors, how they promote tumorigenicity. Like you see here, there is a ionizing radiation that lead to the translocations. Chemicals like chemotherapy usually cause point mutations. So if I tell you about what are the factors which are causing the initiation of a cancer, So that you say the radiation, so what radiation does what? Is basically responsible for translocation. And then if we say the chemotherapy induced cancer, that's what it does. Basically, it does point mutation, predominant phenomena, and that lead to the cancer initiation. The retrovirus genome, basically the genome incorporates in the host cell genome, and then lead to the cancer initiation. So these are the ways to know the this thing. And if you see here the proto-oncogene conversion into the oncogene, apart from a virus infection, the oncogenes also I've told you the regarding the same thing, the chemical ionizing radiation, changing the normal proto-oncogene of a mammalian cells into oncogene, and overexpression and development of a cancer. There are three principles mechanism by which proto-oncogene can be activated to produce the malignant cell. This is very, very important slide. And we should focus that what prevails this proto-oncogene to become an oncogene, because normally it does not happen. Usually the people born with a proto-oncogene and die with a proto-oncogene. But what happens in between, those who have cancer, uh, first is the point mutation. It can occur, and the point mutation is more commonly occurred with the chemotherapy agent. Can occur changing the single base pair, which subsequently produce a protein with amino acids change. I mean, different amino acid, altered amino acid change with the protein happens development. And for example, the point mutation in a KRAS gene found mostly in the patients suffering from the colorectal cancer, colon cancer, pancreatic cancer, lung cancer, thyroid cancer. So these are the point mutation. What is chromosomal reassignment and chromosomal translocation? I told you the chromosomal translocation usually happens because of some ionizing radiation. So often placing a proto-oncogene like translocation has been between the chromosome, like some, for example, I'm telling you the chromosomes 2 is exchanging with the chromosome 8, a segment is responsible for the CMYC oncogene activation, and that happens in the Burkitt lymphoma is a proven fact. The third things, the gene amplification, in which many extra copies of a proto-oncogene is formed in cells associated with the activation of oncogene. In these cells, like example, the gene amplification NMYC is found in a neuroblastoma. So these are the three important points by which the proto-oncogene is developed into the oncogene. The critical features of a oncogene to understand, usually they act in a dominant fashion. That means the presence of a single copy of activated oncogene is sufficient to produce the transformation into the cells for overproliferation, metastasis, and I mean all the features of a cancer initiation. When oncogene is a dominant portion, means dominate means autosomal, likely you must have understand about the dominate, autosomal recessive. So what happens here, if with the both the chromosegments having the altered gene, then it is bound to happen, that's a cancer initiation. But in case of oncogene, a dominant class, when a single segment of a chromosome is having an oncogene which is sufficient to produce this cancer initiation. That is the philosophy which this is I just want to convey you. The development of a cancer oncogene activation occur by mainly two ways. First is the transcription dysregulation, other is gene fusion. So what is transcription dysregulation? In which the oncogene overexpressed and several gene function which happens as a result of incorrect expression of oncogene. Many example of first type like follicular carcinoma in BCL2 gene is dysregulated by juxtaposition, meant the change in a position to immunoglobulin enhancement. So that is the transcription null dysregulation. So, I mean, I'm repeating the terms BCL2. What is BCL2? If somebody asks you, because this gene is discovered in the B cell lymphoma, that is why the name was found in the B cell, BCL. So, second is the gene fusion. Where is gene fusion occur? Was found like in a chronic myeloid leukemia in which APL oncogene fuses with the BCR gene. What is BCR means breakpoint cluster region. With the ABL oncogene fuses with the BCR gene. As a result, as a symmetrical translocation happens between the chromosome 9 and 22 that lead to the development of the chronic myeloid leukemias. So we have a beautiful drug designed for that is the imatinib, which hits on this. So that's that is the help of a by the help of a molecular oncology, you design the drug. The tumor suppression gene, it was discovered in 1970 that tumorigenicity of some tumor cells could be suppressed by the hybridization of a normal cells, because the normal cells having a tumor suppressor genes. The hypothesis was made after that that the normal cells contain a gene that can suppress the neoplastic potential of a tumor cells. So that what is the tumor suppression gene? Tumor suppression gene has been mapped to the specific chromosome, that is chromosome number 11. So the loss of a chromosome 11, a particular segment of a chromosome 11 which is having a p53 gene, a tumor suppressor gene, there are the maximum level of chances that a tumorigenicity should occur in that host. The function of oncogene and tumor suppressor gene. The identification of a gene enclosure in human cancer quickly lead to understanding of function of these genes. Cells must repeatedly make the decision to divide, differentiate, and undergo the apoptosis. But this out of three, divide, differentiate, undergo the normal cells. But in the cancer cells, they divide, yes, no problem, but they do not differentiate, and then also they do not go for apoptosis. All those outcome affect the cell numbers, so it is not surprising that one or another these another of these decisions regarding the above mentioned points. These pathways are primary target of oncogene or a tumor suppressor gene. So let us talk about the telomere and its cancer relation. The telomere basically is a segment of a chromosome which lies in the terminal part of a chromosome, and the mammalian telomere consists of a sequence of TTA triple G TAGA that ranges the length from 1.5 to 150 kilobase. Each time when the normal somatic cells divide leads to the terminal end of a telomere cap lost. So that becomes shortening after division, and then after division, there is a significant shortening after the certain point. And when the significant shortening happens after the certain time, that is the time when the cell goes for a apoptotic death, normal death. So how many times after the 40 to 60 cell division in the normal healthy cells, this vital segment of DNA, that is telomere, are lost. At this time, cell cannot divide and it go for a normal programmed cell death. But this is not happen in the cancer, because the cancer after the each division telomere repairs occur because of help of a telomerase enzyme, which is specifically raised in this particular cancers, particular type of these cancers. So, if you see the age wise, you can beautifully see here a teenager, quite young age, having a significant portion of telomere, but after the lengthening, the significant portion of telomere length is lost in the elderly adult. That is why the cancer is more commonly happens in the elderly rather than the teenage, apart from the some hematological cancer. So you see here the segment of a DNA is the telomere ta ga t a a g g g a t t c c c t. This is a complement component, the cytosine, thymine, adenosine, guanosine. This picture beautifully explains you the telomere significance. So now what is the molecular clock? So the telomere length is being described as a molecular clock for the cell cycle. The stem cells like a bone marrow uh, or cells, some GI tract, mucosa, epithelial lining, as well as some cancer cells avoid this process of a telomere shortening after the division, cell division, by activating the enzyme telomerase. This enzyme is basically a reverse transcriptase enzyme that polymerizes the segment of a telomere, that is consist of TAGA. Repeat offset of this map makes the cells immortal. That is the important thing. And so the cancer cells go for uncontrolled proliferation, and these cells shall not go for a normal routine death. The same thing portion of the telomere cells, and the centromere is in the center, and the DNA, and then a region of a repeated nucleotide sequence at which the chromosomes, telomere. Now let us talk about certain in the depth the signal transduction. What is signal transduction? It is to provide the information to the nucleus regarding the environment outside the cells. The information from outside of the cell goes inside the cell, that is transduction. Basically, signal transduction term used to describe the flow of information from cell's outer membrane to the cytoplasm up to the nucleus in form of various signals. That signals is what are these signals? Cytokinins, low molecular weight hormone, growth factors, and other protein that arrived at the outer plasma membrane can enter these cells via two principal mechanisms. Cell surface receptors that are present on all around the plasma membrane of a cell, and the second is selectively bind the individual cytokinins and a hormone. These receptor molecules transduce the signal from extracellular domain to cytoplasmic domain and lead to activation of enzyme site protein kinase. Now the things start from here. Some extracellular environment agent can migrate across the plasma membrane cells and reach up to the nucleus directly acting on the DNA. So in both the conditions, ultimate information is of a signal. It has to reach to the nucleus level, thus altering the cellular behavior by altering the nuclear behavior. So one important signal transduction pathway involves the GTP binding protein. Include the RAS family. These act as a molecular switch for a various physiological protein that regulate the cell growth. RAS is a key regulator for cell growth in all eukaryotic cells. Mutation in that lead to the various cancers. Almost 30% of all human cancers contain an active mutation in the RAS. RAS is the highest frequency found in the carcinoma colon, CA lung, CA thyroid, and pancreatic carcinoma. So that an anti